So today we are going to discuss the Latin scale design, which is one of the experimental designs. And in the previous lecture, we had studied the basic concepts of experimental designs, that what is meant by an experimental design, what are the different components of experimental design, and what are the principles of experimentation. And we also discussed that there are three basic types of the experimental designs that are used in field research. The first one is CRD, which is the simplest type of the experimental design, in which the only significant source of variation is the treatment, which is source of variation of our interest, and that one is used under lab conditions mostly. And then we had discussed RCBD, which is randomized complete block design, and RCBD is that in which the significant sources of variation are 2. And if we have more than 2 significant sources of variation, then we are going to use the Latin scale design. So RCBD minimizes the contribution of one source of variation other than treatment. As we discussed in the previous lecture, that if we have a source of variation which is unavoidable and that could have the significant effect or that could have a noticeable effect on our response variable, so we need to take measures to control the effect of that variable. And we saw that how we can do the blocking and how we can make blocks to reduce the effect or to calculate the effect of that source of variation. And these blocks are made in the direction of the gradient of that source of variation. So RCBD is good in the field research where mostly we have one significant source of variation other than the treatments. So the Latin scale design can handle 2 sources of variation that occur in a gradient. So RCBD can handle only one source of variation other than treatment, while the Latin scale design can handle 2 sources of variation that occur in a gradient in the field. And every treatment occurs only once in each row and column of the field, which is designed under Latin scale design. For example, we have a field in which there is a river on one side and there is a road on another side. So we are going to see that if we have 2 significant sources of variation, then how we are going to handle those sources of variation through the Latin scale design. So here we are taking the example of a field which has 2 sources of variation other than treatment. One is the river, which is on one side, and the other source of variation other than treatment is the road. So both river and road, they may have effect on the growth of the plants. So therefore, in order to control these sources of variation, we need to design our experiment in such a way that their effect can be calculated in blocks so that we can have a separate calculation for their effect and their effect can be controlled, right? So let's see, here we have our field. So in the field, you can see that there are different scales, and let's see how these scales are made. So we have river on one side and there is a river gradient. So the effect of river is maximum in that side of the field which is near the river, and it decreases away from the river. So the effect of river, the effect of this source of variation is present in the form of a gradient. So what we can do is, as we learned in RCBD, that we can make the blocks. So we can make the blocks according to this river gradient, and here we can make these columns, and you can see that the effect of the river is maximum in column number 1 or the block number 1, and then the effect of the river is less in column number 2 or block number 2 according to river than 1, and then we can see that there is a decrease in the effect of river as we move to block number 4 according to the river gradient. So we have these 4 columns or these 4 blocks, and we have divided the effect of river gradient in these blocks. So this is one source of variation. Now let's see, we have another source of variation which is road on one side, and we can see that the effect of this road is also present in the form of a gradient, because the effects of the road on the growth of the plants is going to be maximum in the plants growing near the road, and it will decrease as we move away from the road. So this road effect, this road source of variation, it is also present in the form of a gradient. So what we are going to do with that is we are going to make blocks according to the road gradient as well. So we have these 4 blocks. The block number 1 has got the maximum effect of the road gradient, and this is presented in row number 1. And then we have row number 2, which is the block number 2 for the road gradient. Then we have row number 3, which is the block number 3 for road gradient. And then we have the block number 4, which is the column number 4 for the road gradient. And we can see that the effect of the road gradient is minimum in this block. So here we are going to make 4 blocks for the river gradient and 4 blocks for the road gradient. So this is how we are going to have a square design, and hence the name of this design is the square design. This is a Latin square design because in this case the equal number of blocks are made for both of the gradients. So in the Latin square design we can have a maximum of only 16 plots, right? So it can have less than 16, but any number of squares. Like if we make 3 blocks, then this is going to be 3 into 3, 9. So at maximum we can have only 16 plots. So this is the limitation of Latin square design, that you can have only 16 plots in that. So at maximum you can have 4 types of the treatments, because each treatment appears only once in row and once in column. So each column is going to have only one copy of each treatment. So if you have 4 treatments, then each of the treatments is present in a single copy in each of the columns. And if we have 4 treatments, then each of the treatments is going to be repeated only once in each row. So here you can see that we have a perfect square design with 4 blocks for the river gradient and 4 blocks for the road gradient. And the number of treatments are also 4. So in the first column, as you can see, that the first cell is D. So the application of the treatment is D. So the treatment applied in this cell is D, and this lies into the column number 1 and row number 1. And then we have the other one, which is A. So A is present in column number 1 and column number 2 and row number 1, and this one has got the treatment A, and so on. So in this you can see that the treatments are repeated only once in each row and only once in each column. So we have 4 blocks for river gradient and 4 blocks for the road gradient. So we have the square design of 16 plots and we have 4 treatments. So each treatment is repeated 4 times, but this is repeated only once in each row and only once in each column. So this is how we are going to make this design. So this is the basic design of the Latin square design. So we can take the example, and the example is effects of fertilizer dose on the fresh weight of plants. And the experimental design is the Latin square design that we are going to use. The independent variable are the fertilizer doses, and the treatments are different doses of fertilizer. And in this example we have 4 treatments, A, B, C, and D. And the dependent variable is our response variable, which is the fresh weight of plants. So this is the experiment that has been planned. So here we have the results of the experiment, and you can see that these are the values of the fresh weight of plants under different doses of fertilizer. So here you can see that we have 16 plants and we have 16 observations, and these observations are according to the plots, according to the blocks that are made according to the river and the road gradient. So these are the observations we have. So we are done with the 2 phases. Remember, what are the phases, or what are the components of the experimental design? We have to plan an experiment to obtain and appropriate data. So we planned an experiment and the experiment is executed and we have the data. So now data is obtained. So what is the next step? We have to draw inference out of that data, and for that purpose we have to apply the statistical analysis, right? So the next step is to do the statistical analysis on this data. So the kind of statistical analysis we are going to use for this data is Anova, and here we have the table for Anova calculation. And as you can see that this table has got all those 16 observations. Then we have the row totals and we have the column totals. So what do these row totals and the column totals depict? They are according to the gradients. So they represent the gradients. The row totals, they represent the road gradient, and the column totals, they represent the river gradient. So the column number 1, which has the sum of 211.46, so that represents the result of column number 1, which is the river gradient 1, and it has all the 4 gradients of road. And then we have the column total, which is 204.14. So this is the sum of column number 2, which is our gradient number 2 of the river, and it also has got all the 4 treatments and all the 4 gradients of the road. Then we have column number 3, which is our gradient number 3 of the river. This value is 194.79, and it has got all the 4 treatments and all the 4 gradients of the road. And then we have 183.59, which is the sum of column number 4, which is the 4th gradient, 4th block of the river gradient, and it has got all the 4 treatments and all the 4 gradients of the road. And then if we move on to the roads, so the total of first row is 200.76. So this first row belongs to the first gradient of the road, and you can see that it has got all the treatments and it has got all the gradients of the river. Then we have the next row, which is 199.27. So this is the total of the second row, and the second row is the second gradient of the road, and it has got all the 4 treatments and all the 4 gradients of river. Then we have 196.05. So this is the total of row number 3, and row number 3 is our gradient number 3 of the road, and it has got all the 4 treatments and all the 4 gradients of the river. And then we have 197.9, and this is the total of row number 4, and the row number 4 is the gradient number 4 of the road, and it also has got all the 4 treatments and all the 4 gradients of the river. So this is how we have obtained our values. And then we have the total values, the summation x total and the summation x square total. But you can see that we don't have any totals for the treatments, which are in fact our main source of interest, right? So what do we do for the treatments? The treatment observations are listed separately, and here you can see we have the totals for different treatments. Treatment A, the total is 250.1, treatment B, the total is 218.06, the treatment C, the total is 204.89, and the treatment D, the total is 120.93. So here we have the values for the treatments. Now remember that what is the rule of Anova? That we have to figure out that what are the components that contribute to total variability. So in this case, total variability is because of the variability due to treatment, of course, and it is due to variability due to gradient A, which is our road gradient, and it is because of the gradient B, which is our river gradient, and it is due to the experimental error. So for the sake of simplicity, we can designate total by capital T small o, treatment by small t, gradient A by capital A, gradient B by capital B, and experimental error by small e. And the gradient A is our road gradient, which is presented in rows, and gradient B is our river gradient, which is presented in columns. Now we have to calculate the correction term as we move on to the Anova. So this is the formula for correction term, summation x t o whole square divided by the total number of observations. So what is the total sum of observations? This is 793.98. We take the square and divide it by 16. So we get the correction term as 39,400.265. So this is the value of our correction term. Now we have to calculate the total sum of squares. So the total sum of squares is equal to the summation x t total square minus ct. So remember, what is this term, the summation x square for total? So this is the square of all the individual 16 observations, and then we take their sum. So this is equal to 41,802.61. So we subtract it from the correction term, which is 39,400.265, and we get the value of our total sum of squares, which is 2,402.345. So this is the value for our total sum of squares. Now we have to calculate the sum of squares for treatment, and this is the formula for the calculation of sum of squares of treatment. So we have 4 treatments. So we calculate the summation x whole square divided by the number of observations for each of the 4 treatments. and then we take their sum and subtract it from correction term. So here we have put all those 4 values and we subtract it from correction term, and we get the treatment sum of squares as 2275.77. So this is the value for our treatment sum of squares. Now we calculate the sum of squares for the road gradient, which is presented in rows. So this is gradient A and this is the formula. So we had 4 blocks according to this gradient of rows, which were 4 rows. So we put in the values for each of these rows, take their sum and subtract it from correction term. So here we input the values for these 4 blocks, for these 4 gradients of the road, and we subtract it from the correction term, and we get the value for the sum of squares A, which is the sum of squares due to the road gradient, and this is 3.02. Now we have to do that for the river gradient, and this river gradient was presented in columns. So we take the totals of these columns, take their square divided by number of observations in each of the column, we take their sum, and then we subtract it from the correction term. So we input these values and we get the value of 108.96. So this is the sum of squares of the river gradient, which was present in columns, and this is designated here as B. So now we have calculated the sum of squares total, the sum of squares treatment, the sum of squares for road, and sum of squares for river. And now we have to calculate the sum of squares for error, for the experimental error. So here is the calculation for sum of squares experimental error, and for that purpose we are going to make use of this basic principle of the Anova in this case, which means that the sum of squares total is equal to the sum of squares treatment plus sum of squares due to gradient A, sum of squares due to gradient B, and sum of squares experimental error. So because we have calculated the sum of squares for total, sum of squares for treatment, sum of squares for A, sum of squares for B, so we can work out the value of sum of squares E. So sum of squares E, or the sum of squares experimental error, is equal to the sum of squares total minus the sum of squares treatment, sum of squares A, and sum of squares B. So here we have these values, and we get the value for our sum of squares experimental error, and this value is 14.595. And now we calculate the degree of freedom, and we are done with the calculation of the sum of squares for total, sum of squares for treatment, sum of squares for river gradient, sum of squares for road gradient, and the experimental error sum of squares. So now we have to calculate their degrees of freedom. And first of all, the total degree of freedom, which is the total number of observations, which is 16 in this case. So 16 minus 1, we have 15 degree of freedom for total. Then we have to calculate the treatment degree of freedom. So the number of treatments were 4. So 4 minus 1 is equal to 3. And then the gradient A, and the number of blocks in the gradient A were 4. So 4 minus 1 is 3. And then the gradient B, so the number of blocks in the gradient number B were also 4. So the degree of freedom for this one is also 3. And then we have the error degree of freedom. And for that purpose, we have to multiply n minus 1 and n minus 2. So what is n here? N is the number of columns, which is equal to the number of rows, because this is the square design that we have. So in the square design, we are going to have the same number of columns and the same number of rows. So which is 4 in this case. So 4 minus 1 and 4 minus 2, we multiply them and we get 6. So this is the degree of freedom for error. So now we have calculated the degrees of freedom. Now we calculate the MS, which is the mean sum of squares or variance. And this is the variance or MS for treatment. And this is going to be calculated by dividing the sum of squares treatment by degrees of freedom treatment. So we have 2275.77 divided by 3. And we have 758.59 as the variance or mean sum of squares for treatment. Then we calculate the variance or the mean sum of squares for our first gradient, which is the A gradient, and this is the road gradient. So this is the sum of squares for A gradient divided by degrees of freedom of the A gradient. So this is 3.02 divided by 3, and our variance is 1.007 in this case. Then we are going to calculate the variance or mean sum of squares for our gradient B. And for that purpose, we are going to divide the sum of squares for gradient B by degrees of freedom for gradient B. So 108.96 divided by 3, we get 36.32, which is the variance or the mean sum of squares due to gradient B. And now finally, we are going to calculate the variance due to experimental error. So this is our error or within variance, and this is equal to the sum of squares for error divided by the degree of freedom for error. So we have 14.595 divided by 6, and we get the value 2.433, which is the MS value or the variance for experimental error. So now we have calculated the MS values. So what is going to be our next step? We are going to compare these MS values. And how do we compare these variance values? We calculate the F values. So here we are going to calculate the F value for the treatment, which is our source of variation of interest. So for that purpose, the variance of treatment is going to be divided by variance of experimental error. So 758.59 divided by 2.433, and we have 311.79. So this is our F value for the treatments, and we can see that this value is quite high. It is actually quite higher than 1. So we will see that how much significant this value is. Now we calculate the effect of the gradient A, which is our road gradient. So for that purpose, the MSA, the variance A, is going to be divided by variance error. So we have 1.003 divided by 2.433, and we have the F value, which is less than 1. So this is probably going to be non-significant, right? Because this is less than 1. Then we are going to calculate the effect of our second gradient, which is our river gradient, and in this case, we are going to divide the variance of gradient B by the variance of experimental error. So this is 36.32 divided by 2.433, and we get 14.928. So we can see that this F value is also greater than 1. And now we have to see that whether this is significantly greater than 1 or no. So now we are done with the calculations of Anova. And the next step is to make the Anova table and compare these calculated F values with the table or the critical values to find out that which one of these are significant and which one are non-significant. And here is our Anova table, and you can see that we have the source of variation in the first column, and the sources of variation were treatment and gradient A, gradient B, error, and total. So the degrees of freedom for the treatment and for gradient A and for gradient B are 3 each, and for error it is 6. And the sum of squares for treatment is 2275.77. Then the sum of squares for gradient A is quite low, this is 3.02, and the sum of squares for gradient B is 108.96. And sum of squares for error is 14.595, and the sum of squares total of all these four sum of squares is 2402.35. Then in the next column we have the variances or the mean sum of squares. So for the treatment we have 758.59, for the gradient A we have 1.003, for the gradient B we have 36.32, and for the gradient for the error we have 2.433. Now we have to compare their F values, these mean sum of squares or variances, and we get the F values, and the F values are documented in next column. So we have 311.79 for the treatments and 0.412 for the gradient A, 14.928 for the gradient B. And as expected, we can see that this 311.79 is much greater than 1, because the table value is 4.76 at probability of 0.05, and the degree of freedom of 3 and 6. So we can see that our calculated F value of 311.79, it is greater than the critical value of 4.76 as well as the critical value of 9.78 and the critical value of 23.7 at probability of 0.001. So our probability is less than 0.001, and this means a very highly significant result. So very highly significant effect of treatments on the response variable. Then we see the gradient A, and the gradient A is less than 1, and we can see that this is non-significant because the critical value for the degree of freedom 3 and 6 is 4.76 at probability of 0.05. So therefore, this is going to be non-significant. And then we have the gradient B, and let's see the effect of gradient B. 14.928 is also higher than the critical value at 0.05, which is 4.76. So this is also significant. And let's see at 0.01, the critical value at 0.01 is 9.78, and our calculated F value for gradient B is 14.928, which is higher than this one. So this is highly significant. Let's see for the next level. So at probability of 0.001, the critical value is 23.7, and we can see that the value of our gradient B is not higher than this one. So our gradient B is also exerting a significant effect, but this effect is highly significant. It is not very highly significant. So here we have the Anova table. Now it is time to report the result. And a result for the source of variation of interest, which is our independent variable. So there is very highly significant effect of fertilizer on the fresh weight of plants. So the F value at degrees of freedom 3 and 6 is 311.79, and the probability is less than 0.001. Then we have the result for our gradient A, which is the road gradient, and the effect of road on the fresh weight of plants is non-significant. So the F at degrees of freedom 3 and 6 is 0.412, and the probability is greater than 0.05. Then we have the result for our gradient B, which is our river gradient, and we can see that there is highly significant effect of river on the fresh weight of plants. So F value at degrees of freedom 3 and 6 is 14.928, and probability is less than 0.01. So this is how we do the calculations for Latin scale design. So this is our Latin scale design, and the Latin scale design handles with two additional sources of variation than treatment. So treatment is obviously our main source of variation, our source of variation of interest. But if we are doing experiment in the field and we encounter two other sources of variation that cannot be controlled, so by changing the experimental conditions, so what we have to do for that is we have to calculate their individual effects, and we can do that through making the blocks according to the gradients of those two sources of variation. So we are going to have a squared design, and this square design can have a maximum of 4 treatments, 4 blocks of gradient A and 4 blocks of the gradient B. So it can have a maximum of 16 plots or 16 experimental units. And we saw the results of that, of a hypothetical experiment, and then we calculated the effects of these sources of variation through Anova, and we found out that the effect of our independent variable, which is the fertilizer dose, on our response variable, which is the fresh weight of plants, is very highly significant, and the effect of the road, which was the other source of variation, it is non-significant, and the effect of the river, which was the other source of variation, is highly significant. So this is how we will be able to differentiate between the effects of these sources of variation on the growth of plants. And Latin scale design is our third type of the experimental design. So we have 3 basic types of the experimental designs: RCBD, and in RCBD there is only one major source of variation, which is treatment. So in total we have 2 sources of variation: treatment and the experimental error. Then in RCBD, which is the most widely used design in the field research, we have 3 sources of variation: the treatment, which is the source of variation of our interest, experimental error, which is due to the extraneous factors which are unavoidable, and we have an additional source of variation which is known as the source of variation due to the environmental conditions that we have over there, which cannot be avoided. And we calculate the effect of that source of variation through making blocks. So we have three sources of variation in RCBD: the variation due to treatment, variation due to blocks, and variation due to experimental error. Then we have the Latin scale design, and Latin scale design can handle two sources of variation other than treatment. So in this design, we are going to have four sources of variation: treatment, which is the source of variation of our interest, and then we are going to have the source of variation due to some other factor which is present in the field, which is not of our interest, but it is present in the field. So we make the blocks for calculating the effect of that gradient, and then that source of variation according to its gradients. And then we have another source of variation, and we are going to have this calculated through making the gradients or blocks for that source of variation. And then we have the fourth source of variation, which is the experimental error. So in Latin scale design, we have four sources of variation, and we are able to calculate their effects through Anova. So this is all about the experimental designs, introduction to experimental designs.